Are we live? Hello students, welcome to Vedantu Neet English. Let me know in the chats if we are audible and clearly visible, and you can also see the presentation behind us. All of you, quickly.
Yes, ma'am. We are live. Yes.
We are live? Okay,
Yes.
awesome. Awesome,
Students,
awesome.
how is everyone doing? Quickly let us know in the chat. How is the preparation coming along? And students, if you do not know us, if you're here for the first time, this is Basparaj sir.
This is Gopika ma'am.
We are your
Biology
biology
master
master
teachers.
teachers. Welcome to Vedantu Neet English students. Students, there's one question we want to ask you. That is,
Are you ready to master?
your NCERT. That is, we'll be doing every single line of NCERT. We have already covered. Now we'll be doing the every in-text
Text questions. Only that?
No, ma'am. We promised you we'll be doing all the bold words as well as the bracket words. We'll also be doing all the scientists as well as
Disorders.
everything. So students, the question is now, are you ready to master your NCERT? Not just by the superficial, but in detail. Not even missing every single diagram, nothing. Nothing should be missed out. So are you ready? Let us know in the chat.
Because we have done the basics, like making the concepts clear, but now is the time for you to target 360 out of 360 in biology, because most of you think that, I can score good in bio, so I can do a little, like here and there I can miss physics and chemistry a little.
Yeah.
For that, you need to know every single word of biology, every single word of NCERT. So that is what this
When
journey
we
is going
say
to be.
every single word, we mean every single
single
word. That is every single table, graph, diagram, the legend of a diagram where questions have actually come from. So every single thing is very important, and we are here to make sure that you do not miss out on any word or any line of NCERT. So, ma'am,
Chalo, let's start.
ma'am.
Are you ready? So all of you, sir, please check if anyone is bunking the class. Quickly check the attendance.
Yes, ma'am. Then quickly, everyone, mark your attendance, and we'll start.
Gopika ma'am will be really mad if you bunk the class. Okay? Yes, come on, all of you smash the like button. That much you can do, because again, we are spending a Sunday with you people. Okay, chalo, let's start with the first thing, students. Biological classification. Open your NCERT with me and we will try to solve it. Okay? So the first in-text question means what? That means it has come in between the paragraph. Maybe the question is a part of the paragraph, or it could be a general question, and MCQs can be framed with that. This is what is in-text question. Okay? So the first question says, would you call virus living or non-living? I want the answers in the chat when I'm explaining as well. So students, the question is, will you call virus living or non-living? The answer is that the virus is somewhere between living and non-living. Why is that? Because if I want to call something living, it has to grow and it has to reproduce. Now the question comes, do this too happen by the virus? No, they do not do it. But when they enter into the host body, with the help of the host energy, they will do reproduction, right? They will do reproduction. So, do you understand this? I cannot call it living because they do not grow or reproduce, but when they enter the host organism, they do reproduce. So, living or virus comes somewhere between living and non-living. This is the answer for this in-text question. Is this clear? Yes, very good. They are a connecting link between your living and non-living.
Right.
Awesome.
Ma'am, let me start botany little, and then we can
sure.
have you, ma'am.
Okay.
So students, let's start with the basic botany now. Ma'am, pen. So students, next question is going to be from the cell, the unit of life. Let me know in the chat if you're able to see the question clearly. If there is some blurriness, let me know in the chat right now. Let's read the question. What is the question now? The question says like, can you recollect names of organisms that have more than one nucleus per cell? That is, they have more than one nucleus per cell. This is the first question. Any question? Any answer? Can anyone tell me in the chat? See, all of you know, in the case of humans, our normal RBC does not have any nucleus, but any other cell will have a single nucleus. But in the case of certain protozoans, such as your paramecium, has multinuclear condition. There are certain muscle cells which has multinuclear condition. Then there are also certain algae like Chara, which has multinuclear condition. Yes, there are certain fungus which is multinuclear in nature, that is penicillium as well as rhizopus. So students, are you getting it? There are certain organisms which has multinucleated condition. Now, what about your red blood cells and CO tubes? How many nucleus do you think red blood cells have? The answer is 0. That is, red blood cells will completely remove its nucleus to accommodate more space for hemoglobin. There it is again, students. See, some mature cells even lack nucleus. Hidden line of NCERT. Example, erythrocytes of many mammals and sieve tube cells. So students, remember, the sieve tube cells will not have nucleus. Now quickly tell me in the chat, is the sieve tube cells, is it living or is it dead? Tell me in the chat quickly. The sieve tube cells, is it living or is it dead? And also tell me in the chat, does the sieve tube cells, are they present in the case of gymnosperms? Students, we are connecting chapters now. We are connecting this chapter, this line of NCERT to your plant kingdom, right? To the plant kingdom. So remember students, sieve tube cells is a living tissue. Yes, and it is not found in the case of your gymnosperms. It is exclusive to your angiosperms. Remember that line of NCERT. They can ask you a question from there. The next question goes like, mammals and sieve tube cells of the vascular plants, would you consider these living cells? Students, remember, a living cell is a cell which is able to do life processes. So a cell like your erythrocyte, a cell like your tube cells, they are able to do life process and hence we call them living. Even though they might not have the nucleus, they are still living because they're able to do life processes. They're able to do what? Life processes. Clear? Can we go to the next question, students? Next question is from your biomolecules, the most famous question, students. The most famous question. That is the molecules in the insoluble fraction. Now, what is this insoluble fraction? See, if you know the experiment, when you do the experiment, this is your beaker. This is the muslin cloth. Now, when we add our slurry, some amount of slurry will travel down to the bottom. Now, this part is called as your filtrate. The above part is called as what? Retentate. Yes. Now, students, whatever substance we find in the filtrate is called as your acid soluble fraction. Now, why is it called as acid soluble fraction? Because whatever substance we find in the filtrate is technically soluble in your trichloroacetic acid, and whatever we find in the filtrate is also called as micromolecules. Yes. And what about retentate? What we find in the retentate is called as your macromolecules. Now, students, tell me in the chat, lipid sizes less than 1000 Daltons. Now, if the size is less than 1000 Daltons, does it belong to? Do we find lipids in the filtrate? That is the question. Do you find lipids in the filtrate? Even though the size of the lipids is less than 1000 Daltons, we do not find our lipid in the filtrate. Where do you find lipid? Lipid is mostly found in your retentate. So we find lipid in the retentate. Now, question is why? Why do we find retentate? Even though size is less than 1000 Daltons, why do you find retentate? The answer is very simple, friends. Have you played the chain game when you were kids? The chain. We hold hands and we stand, right? Chain. When suddenly someone comes and they break the chain. Your lipids are like that. They will hold hands together. They will play chain. And when they hold hands together, they form something called as vesicles. Yes, they form something called as vesicles, and these vesicles do not travel down to filtrate. And vesicles are formed in the retentate. All the vesicles will be available in the retentate. Clear? Clear, all of you? Yes. Next question. Next question from your cell cycle. Next question from your cell cycle chapter. Now, students, what is the question here? Oh, the question is this side. How do plants and animals continue to grow all their life? What is the question? The question is, how are the plants and animals able to grow continuously throughout their life? Obviously, the simple answer is going to be cell division. The simplest answer is going to be cell division. That is, every single cell, some of the cells stop dividing, some of the cells stop dividing, but majority of the cells continuously divide. So make sure the particular organism is able to grow, right? Do all cells in the plant divide all the time? What is the answer here? Do all the cells keep on dividing? Do you think sclerenchyma, parenchyma, collenchyma, do they keep dividing? The answer is no. Not all cells keep dividing. There are most of the cells, they get a permanent government job and they're like, no, I do not want to divide any further. I will simply do a fixed function and I will stay quiet. So not all cells divide here. Next question. Do you think all cells continue to divide in all plants and animals? Again, the answer is no here. Can you tell the name and the location of the tissues having cells that divide all their life in higher plants? The answer is there are some cells which keep on dividing. They do not have a home. They are like, they'll keep on roaming throughout the world. You know how some people do not have a house, they keep on roaming, travelers. There are some cells which do not have a permanent function. They'll keep on dividing. That is meristematic cells. Example is your meristematic cells. They keep on dividing. Example is your Ram and Sham. Ram and Sham. Ram, root apical meristem and shoot apical meristem. The shoot apical meristem keep on dividing and the root apical meristem also keeps on dividing. Now, do animals have similar meristematic tissue? The answer is no. The answer is no. We do not have similar meristematic tissue. Some similarities there, but not exactly meristematic tissue. Clear? Some can argue here, sir, liver cells keep on dividing, but they do not. They regenerate. They do not keep on dividing. Okay. Students, is that point clear to all of you? Is that point clear to all of you? Yes. If it is clear to all of you, a quick fire in the chat will make sure I know that every single point is clear to you. Okay. And students, if you have any doubts regarding any of the part in today's lecture, let us know in the comment section and Gopika ma'am and I will personally reply to every single doubt in the comment section for you. Next question, students. What is the next question? Numerical. You have studied the, let me zoom it for you. I can't zoom it, is it? Let me zoom it for you. I can't write now, right?
Write it with the other thing.
You have studied mitosis in onion root tip cells. Yes, we have studied mitosis in onion root tip cells. It has 16 chromosomes. So from the first above equation, we know, okay, which is the one here? This one. This one. So here, what are the information we have? We have here is that 2n is 16. One second. Here 2n is 16 here. Yes, from the above information. 16 chromosomes in each cell. Can you tell how many chromosomes will the cell have at G1 phase after S and M phase? One second. Let me just go to the next one. That will be easier. I think the question is there right here. Better. Now, what is the question saying? You have studied the mitosis in onion root tip cells. That is, the 2n is equal to 16. It has 16 chromosomes in each cell. Now, can you tell how many chromosomes will the cell have at G1 phase, after S phase and M phase? So, students, remember, in G1 phase, is there any reduction of chromosomes? No. The chromosome number will remain the same. Now, what about S phase? Now, remember, students, in the S phase, there is duplication of DNA. There is no increase in chromosome number. There is only increase of chromatid number. Yes, chromosome is like this. There is an increase in chromatid. But are there any increase in chromosomes? The answer is no. So, in the S phase also, chromosome number will remain same. Now, after S phase and after M phase. Now, tell me in the chat, in mitosis, is there any reduction of chromosomes? In mitosis, we never have reduction of chromosomes. The chromosome number will remain the same here. Chromosome number will remain the same. Done. First part of the question is done and dusted. Let's look at the next part. Also, what will be the DNA content of the cell at G1 phase, S phase and G2 phase? Easy question. Listen to me very carefully. In G1 phase, if the DNA content is 2C, at G1 phase, the DNA content is 2C in nature. Now, students, in S phase, if you know in the S phase, there is duplication of DNA content. That is, the 2C will become 4C here. Yes, 2C will become 4C. What about your G2 phase? In G2 phase, again, there is no duplication of DNA content. The DNA content will remain the same after the ES. So this is going to be 4C in nature. It's going to remain 4C as such. Done? Easy question? Done. Next question is from your photosynthesis in higher plants. Can we start? Next question from your photosynthesis in higher plants. The first question is right here. What is the first question? Can you see this paragraph? Have you ever thought what would happen if there were no oxygen to breathe? Can we survive? Are we able to survive without oxygen? The answer is no. So the answer to this question is very simple. That is, all the aerobic organisms, not microorganisms, all the aerobic organisms such as humans, we would not be able to survive. But anaerobic organisms and microorganisms would be able to survive. Easy question, easy answer. Next question, students. Next question is here from your experimental proof. Can you explain how this conclusion would be drawn? So for you to understand, I don't know, I'll show you the thing. So students, this particular question, the context is your above one, that is CO2. Now let's read the question again. That is, CO2 was required for photosynthesis. Can you explain this conclusion? How can this conclusion be drawn? Very simple again, students. If this is your plant, let's do a simple plant here. If this is your plant, this is the leaf. Now take this, we have one plant here, we have one more plant here in a jar, opposite phyllotaxy, right? Now what you need to do is take the first plant, blue. Now completely cover this plant, Done. Covering is done. Now in this plant, what you need to add is, we need to add KOH. In this, nothing is there. So what exactly this KOH will do? The KOH will completely absorb every single CO2 inside the jar. So will there be any CO2 present here? No. So minus CO2 here. Minus CO2. What about here? Here we have plenty of CO2. Carbon dioxide is present in plenty. Carbon dioxide is present. Now after some time, take a leaf. Take a leaf from here and take a leaf from here. Now you need to do a test called as your starch test. That is, test for iodine. Take some iodine, add it on the leaf. If the leaf will turn blue-black in color, it shows that a particular leaf has starch. Now you tell me in the chat, which particular plant, A or B, which plant will show positive for starch? Will there be starch production in plant A? Will there be starch production in plant B? Quickly tell me in the chat. Tell me in the chat, tell me. So students, answer is very simple. That is, your plant A will not have any form of starch. There will not be any form of starch. Only plant B will have starch because there is CO2 is present here. Since CO2 is absent here, there will be no starch formation in your plant A. Clear? Easy question, friends. Easy question. The next question is, how many different ways can you think of to light the candle without disturbing the setup? Now this question is very simple. They might not ask you. The answer is very simple, students. Use some physics. That is, this is concave, right? Okay, same. So take a concave lens and nicely you take an angle and light the candle. Or you can use some kind of spark to light the candle inside the jar without disturbing it. Next question. Clear? Let's go to the next question is here. The question is, can you explain why 12 molecules of water as substrate are used in the equation given above? Let's say the question. Let's say the equation. This is the equation now. Students, can anyone tell me in the chat, why do we require 12 H2O here? Why? Can anyone tell me in the chat? Now don't tell me, sir, chemistry balance in the equation, or don't tell me now. What is the actual reason that there is 12 H2O here? Answer, anyone? Very simple. Tell me in the chat, to make one molecule of glucose, how many times does the dark reaction has to happen? All of you know. For to make one molecule of glucose, the dark reaction has to happen 6 times. Yes, 6 times. Clear. Now in one dark reaction, how many ATPs required? Tell me in the chat. In one dark reaction, there is 3 ATPs required and 2 NADPH is required. Yes. Now let's multiply into 6. 18 ATP is required for one molecule of glucose and 12 NADPH is required. And to make this 12 NADPH, do we require a hydrogen donor? Yes. And the hydrogen donor is going to be your H2O. That's what. To make one molecule of glucose, since we require 12 NADPH, the hydrogen donor comes from your 12 molecules of water. Clear? Easy question? Yes. So this is how you need to explain this particular concept. If you did not know this particular concept, quickly like the video right now and also a green heart in the chat because I want to see some botany love now. Yes, botany love. Next question, students. Next question is, also in other green parts of the plant, can you name some other parts where do you think the photosynthesis may occur? Right? There might be other parts of the plant where photosynthesis might happen. See, we know the majority of the photosynthesis happens in the leaves. But do you think a particular petiole can also take place? Right? A petiole can also make sure that photosynthesis can happen. Example is going to be your Australian acacia. Similarly, if you think a particular stem, phylloclad, a stem can become photosynthetic. Example is going to be your Opuntia as well as your Euphorbia, where a stem can become photosynthetic. All of you know that answer from your morphology chapter. Yes. So those are the different places where a particular other part of leaf can do photosynthesis. There is also some form of roots called as assimilatory roots. Now these assimilatory roots can also do photosynthesis. Okay. Easy question. Next question, students. We have one more question here. Now, one more question is there. What is the question? When do you think the chloroplast will be aligned with their flat surface parallel to the walls? Or when would they be perpendicular to the incident light? That is, arrangement of chloroplast can vary. Now, the main criteria to determine the arrangement of chloroplast is the light intensity. Now students, if there is very low light, now what will happen? If there is very low light, if this is your plant cell, all the chloroplast would be parallel. I'm becoming a little gibberish. All the chloroplast would be parallel. Why? Why do you think they will be parallel? To make sure most of the light is falling. Yes. When there is low light, they'll become parallel to each other. Now, what about when there is very high intensity of light? When there is very high intensity of light in this particular cell, what will happen? All the mesophyll cells would be perpendicular to each other. Yes, they would be perpendicular to each other, and in which all the chloroplast would be scattered around. Now, why is the reason here? The main reason why they're perpendicular here is to make sure there is no photooxidation. Photooxidation can also become, that's why they're perpendicular to each other. Students, concept, you might think the question is very trivial. You might think the question is very easy, but there is a reason why plants are like this. That is why plants are better than animals. Animals are mindless. They don't think at all. Plants are so smart that they make sure they're, undergo different type of modifications. Yes, plants are superior. Next question, students. Next question.
Standard of sunlight into photosynthesis.
Yes. Can you guess which is the most abundant plant pigment in the world? All of you know there is one plant pigment which is called as the universal pigment in plants, which is the pigment which is present in every single plant. That is going to be your chlorophyll A. Chlorophyll A. Clear? Chlorophyll A is a universal pigment. Clear? Next question, students. From the figure 11.3a, where is the 11.3a? 11.3a is here. Okay. 11.3a. Can you determine the wavelength, that is the color of light at which the chlorophyll A shows the maximum absorption? Now tell me in the chat, at what wavelength does the plants actually function? That is, we call it PAR. All of you know what is PAR? Photosynthetically active radiation. Now what is the wavelength range? Tell me in the chat quickly. The wavelength range is 400 to 700 nanometer. All of you know that. 400 to 700 nanometer. But when does the chlorophyll A actually function higher? Tell me in the chat. See, if you look at this graph here, at this point right here, can you see? This point right here and this point right here, we see the maximum absorption of light. That is your blue region as well as the red region. Blue region is approximately your 450 nanometer, and this is around 650 nanometer. 650 nanometer. Clear? 450 as well as 650 nanometer. Next question, students. Does it show another absorption peak at any other wavelength too? If yes, which one? That is, if you look closely enough, students, if you look closely enough, here, right here, this is going to be your 450. But do you see this point here? Can you see this small point here? If you cannot see here, look at your NCERT right now. There is a small peak right here around 400 nanometer. That is the other peak they're talking about. Let's see the next question. By looking at the figure 11.3c, the last one, 11.3c, can you say that there is a complete one-to-one overlap between absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? Is there any overlap? The answer is no. There is no overlap at all. See, if you look closely here, there is overlap to a certain limit, but I'll tell you exactly where the overlap is not happening. Can you see this is your light blue color? The light blue color is your absorption spectrum. Yes, this is your absorption. Now, if you look closely, between 500 to 600 for chlorophyll A, is there any absorption happening here? No. Chlorophyll A is not getting absorbed between 500 to 600 nanometer. But is there any action going on? Is there any photosynthesis happening at that range? The answer is yes. Even though there is no absorption, is there photosynthesis happening? The answer is yes. Can you see the action spectrum? The above one, this is your action spectrum. In the action spectrum, there is photosynthesis happening even though there is no absorption. What does it tell you? It simply tells you that there is some other gundas. See, there is, if you see in the movies, there is one main boss. The main boss is sometimes very lazy to do the work. What does it tell? He has small goons. He'll make sure that small goons go to the different poor vendors and collect the money from them and come. Similarly, here we have small goons. The small goons are called as your auxiliary pigments. These auxiliary pigments are your chlorophyll B, carotenoids and other pigments. Now these pigments, even though they might not do the full function, they will absorb all the light and they'll give it to the chlorophyll A. They might not do the photosynthesis because they do not have the enzymatic action. But they will absorb the light and they will give it to the chlorophyll A, and the chlorophyll A will do the work. Clear?
We prepare them for the exam.
Yeah, like we are not writing your exam, but we are preparing you for the exam here. So this is us. Done. Technically we are gundas now. That's why there is no overlapping, students. That's why there is no overlapping in that particular region. Clear? Next question. Splitting of water. You would have asked, how does the PS2 supply electron continuously? Very easy question, very easy answer. That is, even though it's called as non-cyclic, there is continuous supply of electrons. The answer is given right here, that is photolysis of water. With the help of photolysis of water, there is continuous supply of electrons. Continuous supply of electrons. Clear? Now, what is the other question here? Membrane of thylakoid. See, where are the protons and oxygen formed likely to be released? In the lumen or in the outer side of the membrane? That is, whenever the photolysis of water is happening, what is the fate of this particular protons and what is the fate of this poor oxygen? All of you know. Oxygen is technically the waste product, also called as the byproduct here. Yes, byproduct here. Now this byproduct will be released outside the membrane. Will release the outside the membrane. Now what about the H plus here? Students, if this is your thylakoid and this is where the photosynthesis is happening, all the H plus, this is your photosystem, all the H plus will be sent to the lumen. All the H plus will be sent to the lumen. Now what is the use of this H plus? Now this H plus will be used in the, where? This H plus will be used in your preparation of your ATP, chemiosmotic hypothesis, the potential gradient formation. That is the answer. Clear? Done, done, done. Yes, all questions are done here. Next question, students. Can we hence say that calling the biosynthetic phase as the dark reaction a misnomer? Now, what is the meaning of misnomer? The meaning of misnomer is something which is not true. Now, technically, when you think, if I tell you, imagine if you are in from your class 10th now, if I tell you there is a light reaction and there is a dark reaction, what would you think? Light reaction happens in light, dark reaction happens in darkness. You would think that, right? That is what is called a misnomer here. That is, light reaction will happen in light because it is light dependent reaction. But the dark reaction, students, the dark reaction is called as dark reaction because it does not happen in dark, because it is light independent reaction. It does not require light at all. It's like, no, why will I require light? Oh, I do not require light at all. It is like, I will only use the products of a light reaction. I do not require light. I am light free. Okay, that is the answer here. Next question, students. Now let us ask ourselves the question that was asked by the scientist. Now I'm asking you the question which was asked by the scientist also. Scientists also had the same question. Now what is the question? All of you become scientists now quickly. Who was struggling to understand? Even scientists were struggling. They're like, even I was going to say Isaac Newton, then I realized that Newton is physics. So students, even all the different scientists were also not able to tell. What they were not able to tell? That is, how many carbon atoms would a molecule have which after completing fixing of CO2 would have 3 carbon of PGA? Simple question. They are trying to complicate here. Listen to me very carefully. See, all of you know in your dark reaction, we have RuBp. Now quickly tell me in the chat, RuBp, how many carbon molecules? RuBp? RuBp is your 5 carbon molecule. Yes, it is 5 carbon plus. Now to RuBp, are we adding a molecule of your glucose? So 1 carbon. Now tell me in the chat, what is 5 plus 1? Anyone in the chat can tell me what is 5 plus 1? Full basic, different, full high level maths. 5 plus 1 is your 6. All of you know. That is, they're like 5 plus 1 of so 5 plus 1 is 6 carbon. So all the scientists were so confused. They were actually looking for a 6 carbon compound. They were looking for a 6 carbon compound. But did they find a second compound? No. Yeah. What did they find? They found an imposter. That is, they found a 3 carbon 1 and a 3 carbon 1. And this 3 carbon 1 is nothing but your PGA. That is your PGA. Clear?
5 plus 1 is 51.
5 plus 1 is 51. Nice. Maths also fail now. Next question, students. Well, basic, we're telling so basic, students. This is all concept. We're not even just explaining the question, we're explaining the concept also. Do you realize? Next question. Now, this is a Neet PYQ, students. This directly is a Neet PYQ. This is why you need to study the questions. This is a Neet PYQ. Neet PYQ is directly been asked from your line of your NCERT. That is, work out how many ATP and NADPH molecules will be required to make one molecule of glucose. Students, do you see that? In-text question can directly come as the Neet PYQ. So can you take a risk of not studying in-text questions? You might be like, stupid question, who will read this? But these stupid questions will actually come in your exam and you'll be baffled that time. Okay? What happened?
Shakespeare.
Shakespeare. Through the Calvin pathway. Now what is the answer here? Answer is given right here. That is, to make one molecule of glucose, we require 18 ATP as well as 12 NADPH as well as 12 NADPH. Clear, students? Clear? Now, next question is here. There are a lot of questions from plant physiology. Do you realize? There are so many questions from your class 11th in-text questions. And some of these questions you would have been like, such an easy question, I'll do it somewhere else. But we are here doing all the in-text questions. Do you notice the difference? Okay, what is the question here? Study vertical section of your leaf. They're asking you to be a scientist again. One of the C3 plants and other of a C4 plant. So we have a C3 plant, we have a C4 plant. Now what is the anatomical difference between C3 as well as your C4 plant? It is, do both have same type of mesophils? The answer is no. C3 plant is very stupid. It will do because it will spend its ATP like there is no tomorrow. It is like you go out, take out your GP and keep paying money. Because it will do a process called as your photo respiration will happen in your C3 plants. But C4 plants are very smart. They'll be like, no, they'll not have GP only. They'll not have phone only. They'll uninstall everything. That is, your C4 plants will have a special mechanism through which they will make sure there is no photo respiration. How do they do it? Let's look at the anatomy. If the anatomy which we find in your C4 plant is called as your Kranz anatomy. In a C4 plant, we find the Kranz anatomy here. This is the image of Kranz anatomy. If you take a cross section of your C4 plants right now, this is what you will notice. If you notice here very clearly, this is your vascular bundles. Vascular bundles are completely covered by bundle sheet cells. Bundle sheet cells are covered by your mesophyll cells. This is your Kranz anatomy. Bundle sheet cells completely covered by your mesophyll cells. And we call this wreath-like structure. Wreath-like structure. And by doing so, they can do one major thing. That is, they completely isolate CO2 and oxygen. Clear? So Rubisco cannot make a bond with oxygen here. Next question. Based on the above discussion, you should read it. Okay. Discussion. Can you compare plants showing C3 and C4 pathway? Use the table format given in the table 11.1, students. This table is very, very important. Very important. So many questions have actually come from here, and I'll repeat, students, many questions will come from this table. Many questions will come from this table. So are you ready? Are you ready for this table? Tell me in the chat. Clear? MS Dhoni, yes, all of us love MS Dhoni. Who doesn't like MS Dhoni? I don't know, Virat Kohli also, because RCB fans, sad life we have. No cup, nothing. So students, can we start the table? Can we start the table? Yes. So I have made a table for you. Quickly, let's go through the table. And don't worry, this information, this particular PDF will be available to you on the Telegram channel. So go join the Telegram channel right now. You can that is Vedantu Neet English Telegram channel. Join it right now. The entire PDF will be available there. Okay. Let's start. Here, I have made the PDF for you like this. I have made the entire table in a better manner for you. If you read it, so you can read it. Now, first characteristic. Cell type in which Calvin cycle takes place. So where does the Calvin cycle takes place actually? In the case of your C3 plants, it takes place in the mesophyll cells. But in the case of your C4 plants, C4 plants, the Calvin cycle will happen in your bundle sheet. It happens in the bundle sheet. Clear? Next one. Cell type in which the initial carboxylation reaction occurs. Carboxylation, the initial fixation in the case of your C3 plants is mesophyll because it only has mesophyll. But the initial fixation in the case of your C4 plants will happen in the mesophyll. So students, remember, initial fixation of CO2 is happening in mesophyll cells, but the Calvin cycle is happening in the bundle sheet. Two different places. That is the beauty of your C4 plants. Clear? Now, how many cell types does the leaf have that fix CO2? In the case of your C3 plants, there is only one leaf type, that is mesophyll cells. In the case of your C4 plants, there are two types, that is mesophyll cells as well as bundle sheet cells. Okay. Then, which is the primary CO2 acceptor? If you remember, in your dark cycle, also called as your C3 pathway, the primary CO2 acceptor is going to be Rubisco. It's RUBP. It's going to be your RUBP. But in the case of your C4 plants, it's going to be your phosphoenol pyruvate, that is PEP. So PEP will accept the CO2. And students, tell me in the chat, where do we find PEP? Do we find PEP in the mesophyll cells or do we find it in the bundle sheet cells? Tell me in the chat quickly. So all of you know the initial fixation is happening in your mesophyll cells. So PEP will be present in your mesophyll cells. Students, we are actually summarizing your entire C3 cycle and C4 cycle. Remember that. Next question. The number of carbons in the primary CO2 acceptor. Here RUBP is 5. PEP is going to be 3 carbon compound. Clear? Next one. Which is the primary CO2 fixation product? They're asking you the product now. So when RUBP is attached to the carbon CO2, we get the PGA, which is a 3 carbon compound, and that is why it's called as C3 plants. That is why it's called as your C3 plants. Similarly, when your PEP, phosphoenol pyruvate, joins with the CO2, the first stable product in the case of your C4 plants is going to be your OAA, oxaloacetic acid. That is your 4 carbon compound. Now, does the plant have Rubisco? The answer is yes. Both the plants have Rubisco. Does the plant have peptidase enzyme? Students, remember, peptidase enzyme will help conversion of PEP plus CO2. Now, where do you find PEP? Phosphoenol pyruvate is only found in your C4 plants. So, peptidase will not be present in your C3 plants. Peptidase will only be present in your C4 plants, not present in your C3. Which cell the plant have Rubisco? Here mesophyll cells, here bundle sheet cells. Why? Because inside the bundle sheet cells itself, the Calvin cycle is happening. That is why bundle sheet cells, Rubisco is present. Now tell me in the chat, do you find Rubisco enzyme in the mesophyll cells in your C4 plants? Tell me in the chat, do you find Rubisco enzyme? Do you find Rubisco enzyme in the mesophyll cells of C4 plants? Mesophyll cells of C4 plants. Do you find Rubisco? Tell me in the chat right now quickly. Next question. CO2 fixation rate under high light conditions, medium, high light conditions will happen more. Whether photorespiration is present at low light intensities? No. Remember students, photorespiration will not be present at all in C4 plants. There is absolutely no photorespiration. But here there is negligible because low light condition, C3 plants also will function very properly. Similarly, whether photorespiration is present at high light temperatures? Yes. In your C3 plants, they'll give up. When there is very high light, they'll undergo photorespiration. But here, no. It's the alpha. This is your alpha Chad. They're like, no. Whatever happens, I will not move. Now here we have whether photorespiration would happen at low CO2 concentration? Yes. No. Whether photorespiration would be present at high CO2 concentration? No. Because high CO2 is better, no. Better for the plants. Not happening. It's not happening at all here. Optimum temperature is going to be 20-25 degrees Celsius, 30 to 34 degrees Celsius. Example is going to be wheat and rice. Example is going to be maize, sugarcane and sorghum. Maize, sugarcane and sorghum. Done? Clear? Next point, next question students. Now, can you then calculate how many ATP molecules are directly synthesized in this pathway from one glucose molecule? That they're asking about glycolysis here. They're asking you about glycolysis here. Now tell me, how many ATPs are directly produced? So we have here 2 ATP here. We have 2 ATP here, right? 2 ATP here and 2 ATP here. And that is in total of 4 ATP are produced in your payoff stage. 4 ATP are produced in your payoff stage. What about in your preparatory stage? In preparatory stage, 2 ATP are used up. So, minus 2 ATP. Minus 2 ATP. So, what is the net ATP gain? Just ATP. They're not talking about NADH. They're only talking about ATP. Directly ATP. Okay? Substrate level phosphorylation. The answer is going to be 2 ATP. If they ask you how many ATPs are directly generated from your glucose, it's 2 ATP. Okay? 2 ATP. This is net. Net. Next question. What is the net ATP that is synthesized? Calculate how many ATP are synthesized and deduct the number of ATP utilized during glycolysis. Now, when one molecule of glucose is fermented to alcohol or lactic acid. Now they're asking you what is the net ATP gain in the case of your alcoholic fermentation as well as your lactic acid fermentation. What does it tell me? So we just learned the net ATP gain, right? The net ATP gain in your glycolysis is going to be 2 ATP, right? Now all of you know, in the case of your alcoholic fermentation as well as lactic acid fermentation, NAD plus is getting used here. Can you see? NAD plus is getting here. NADH is getting used here. NADH is getting used here. So can we account for the NADH here in the lactic acid and alcoholic fermentation? No. So what is the net ATP gain in your alcoholic fermentation? It's going to be remain as 2 ATP. So what is the answer here? 2 ATP. So remember students, this can be asked as a question, direct question. What is the net ATP gain in the case of your alcoholic as well as your lactic acid fermentation? Answer is 2 ATP. Okay. Next question. What then would be the maximum concentration of alcohol in the beverages that are naturally fermented? The answer is very simple. It's 13%. It is 13%. It is given later on in the chapter. It is 13%. Now, what if the alcohol percentage exceeds the 13%? That is called as your alcoholic poisoning. So naturally, it is 13%. So all of you know the whiskey, rum, all that is higher concentration, vodka, higher concentration of alcohol. How do they get it? The process of distillation. Okay, 13% is naturally occurring. Next question. What is the role of NAD plus? What is the role of NAD plus and FADH2 that is synthesized here? Very easy. NADH and FADH2 are sent to the next village. That is your electron transport chain. They are used in your electron transport chain as a hydrogen donor and electron donor. Next is your plant growth regulators. Students, are you noticing how many questions have actually come from your in-text questions? So many PYQs have actually come from in-text questions. So this is the questions which you need to solve also apart from your PYQs, and we are here to make sure this is happening. Okay. Next question. Therefore, for example, expansion of leaf is growth. How would you describe swelling of piece of wood when placed in water? Simple definition, students. The definition of growth is irreversible change. Now you tell me, if I take a piece of wood and you keep it in water, it swells. But you keep it out of water for some time, will it shrink again? Yes, it will shrink again. So can we call it growth? No, sir. We cannot call it growth. Next question. What would happen if the meristem ceases to divide? Does this ever happen? Students, this does happen in some hormonal cases. In plants also, it happens. If it completely ceases, the plant will not show any type of growth. The seed itself will not germinate. Because you remember, for a seed to germinate itself, we need hormones. We need the meristem to grow, the primary meristem to grow, right? Because we know the primary meristem, students, all of us know, right? The primary meristem is responsible for the primary growth of plant. Primary growth of plant. If the primary meristem is not functioning, will there be primary growth of the plant? No. There will be no primary growth of plant. So it is required. Clear? Next question. Can you think of more similar examples? What kind of curve can you expect in a tree showing seasonal activities? Students, we have two growth models, right? We have two growth models. One is your logistic growth. One is your arithmetic growth. Arithmetic growth can be found where? Tell me in the chat quickly. Where do you find arithmetic growth? Example? Example is going to be your root elongation. Arithmetic growth. Now, what about geometric growth? Geometric growth can be seen in most of the cases of your plants. That is, we have a lag phase, log phase, and a symptotic line. Yes. Now, this particular type of growth called as your geometric growth can be seen in your most of the plants. The example is going to be here. Seasonal activities of plant is also your type of your geometric growth. Okay, geometric growth. Now, next question is here. However, one of them show much higher relative growth rate. Which one and why? They're asking you which plant is showing more relative growth. There are two types of growth, of you know. One is absolute growth. One is your relative growth. Now, quickly tell me in the chat what is the difference here. Tell me in the chat. Let me drink some water. See, all of you know in your relative growth, absolute and relative growth rates. Now, one type of growth is only showing you the actual increase, relative, absolute growth. In your relative growth, it is showing growth per unit time. Yes, it is showing your growth per unit time. Now, where do you find much more relative growth? That the formula for relative growth is what? The formula for relative growth is going to be final minus initial. What is the final here? n minus 5 by 5. That is going to be your into 100. Into 100, that is 100% here. Now, what about here? It is 55 minus 50 divided by 50. That is 5 by 50. That's around 10%. It's around 10%. So, where do you find more relative growth? The answer is going to be your A. Here. Next question. How would you describe a tumor? What would you call the parenchyma cells that are made to divide under controlled laboratory conditions during the plant tissue culture. Students, remember, a parenchyma cell is a normal cell. If it is made to undergo differentiation, it is called as what? Answer I told you. It is a differentiation. That is, it is undergoing differentiation. That is the second answer. The first answer, what do you call a tumor? Tumor is basically a type of mass resulting of uncontrolled growth. Resulting of uncontrolled growth. Clear? That is the end of it. That is the end of it. Students, with that, my duty is not done here, half done. I'll come back again for your other chapters. Until then, let's see Gopika man's face for some time. So, finally.
Is it possible to click on that? All right. Yeah. No, it's there. Okay. Students ready? Let's start. Neural control and coordination. Audible, visible. Everything is good. I think they're still telling the answer, no?
Yeah.
One second. Okay. Hello. Yes. So students, neural control and coordination. Let's go. Okay. So first question here speaks about, do you know why the membrane of a neuron is polarized? The question is, do you know why the membrane of a neuron is polarized? This is a question, very easy question. Students, we all know that the neuron, the polarization, or they're coming into resting potential, action potential, all of this is based on the differences. Yes or no? Right? Differences. So, look here, if this is how your membrane is going to look, so we will have your positive because of the sodium, right? Sodium ions on the top. That is the reason why we have the positive side. Why do we have negative? Negative is mainly because of the negatively charged proteins, but then we also have potassium. Right, potassium and negatively charged proteins are the reason why there is a, so these two people will create a potential difference, right? They will create a potential difference because of which your neuron is always polarized. Got it? Yes. Got it? Because of the potential difference. And who creates this potential difference? These ions create the potential difference, and we all know that. Look here, all of you. If you see here, the question here only, the answer is clearly here. See, different types of ion channels are present on the neural membrane. These ion channels are selectively permeable to different ions. When a neuron is not conducting any impulse, it is called as resting potential, okay? And the axonal membrane is comparatively more permeable. Very important. The axonal membrane is more permeable to potassium ions and nearly impermeable to sodium ions. So this creates a difference. Because of this difference, it is polarized. Are you all with me? Yes? Got it. Good. Okay, chalo, let's go. Okay, next question is, do you know how the presynaptic neuron transmits an impulse across a synaptic cleft to a postsynaptic neuron? All of you know there is a postsynaptic, and between there is a synaptic cleft or a synaptic gap. A gap is there between them. Now, all of you tell me in the chats, how does an impulse go from a presynaptic cleft to a postsynaptic cleft, or in simple words, from one neuron to another neuron? All of you in the chats. I think the chats are delayed here a little. So anyway, tell me in the chats. Now, all of you see here, you can see a presynaptic neuron, that means the first neuron, and then you can see a postsynaptic neuron. Got it? So the answer is again in the question only, how it will be transformed with the help of something called as your neurotransmitter. Okay, neurotransmitter. Neurotransmitters are chemicals. Inside that there could be any kind of chemical, right? It could be acetylcholine. Okay, any of it. ACH could be there. Now these neurotransmitters will open up. Okay, when the impulse will come, they open up, and who will be waiting to receive them? The postsynaptic neuron will have receptors which will accept this neurotransmitter, and that is how a synapse is formed, right? Acetylcholine. Yes, very good. Okay. So Presynaptic to postsynaptic with the help of a neurotransmitter. Neurotransmitter. If you did not see the answer is here. Chemicals called as neurotransmitters are involved. Got it? Answer. Sometimes answer is there in the in-text question. Sometimes it is not there. Okay? Chalo, let's start class 12th. Ready for class 12th? I cannot see the chats. Yeah, it's very disturbing that I cannot see the chats. 1 second. Let me see. Yes, I think all of you are telling the answers here. Is there a delay? There was a delay. Now it's fine. I think now it's fine. 1 second. Yes, delay is gone, right? There was a delay, but the delay is gone. Okay, students, I'll keep the chats here because their chats are lagging. Okay. Yes. Okay, let's start sexual reproduction in flowering plants. One of my favorite topics. Sir took that slide changer and went off. Oh my God. Okay. Chalo. The first question. Find out the names of 5 more flowers that are used in social and cultural celebrations in your family. Okay. Have you heard about floriculture? What does it refer to? If the question is not visible, don't worry. Here the question is there. Okay. So they're asking, list any 5 flowers of ornamental value. So all of you tell me in the chats, which are the 5 flowers you know of ornamental value that we use for puja, for marriage, for whatever it may be. What are the flowers that we use? Very simple. Jasmine, marigold, rose, tulip, orchids, your snake plants. We use a lot of these plants, right? For decoration, to keep in our home, all this. Okay. Now the question is, have you heard about floriculture? Okay. Have you heard about floriculture? So students, floriculture is a part of your horticulture. In this, what happened? We cultivate or we grow some set of flowers which are actually of some use. Okay. It could be cultural. Okay, cultural use, like in the sense for your marriages, puja and all of that we use, and it has some ornamental value. Okay, it has some ornamental value. Is this clear? Floriculture is a part of horticulture where we cultivate some floral ornamental flowers. Clear? Yes. Okay, marigold, rose, jasmine, snake plant, tulip, orchid and many more, right? Many more. Fine. Next question. Can you name the 2 parts in the flower in which 2 most important units of sexual reproduction develops? Very easy question. Who are the essential part of the flower? Who is the essential part of the flower? Essential part of the flower? In the chats, all of you. Essential part of the flower. Quick. Yes. They are busy in their life.
There's a lag, ma'am.
There's a lag, no? No, but here, I think it stopped for a minute. Yes, awesome. Andrecium and gynoecium, right? So the sexual reproduction is only happening because of your andrecium, and andrecium has what? Stamen, right? Stamen, anther and filament, and then you have your gynoecium. Okay, gynoecium, that is your stigma, style and ovary. Stigma, style and ovary. And ovary. That's all. Easy question. Next one. Okay, can you name the parts? Done. Okay, look here. Can you think of, very important question, okay? Can you think of how tapetal cells could become binucleate? First question. Second question. What would be the ploidy of the cells of a tetrad, of a microspore tetrad? What is a ploidy? Very interesting, very good question. See. Can you think of how tapetal cells are binucleate? Students, this is mainly because tapetal cells will undergo, they have large nucleus. Because they have large nucleus, they first undergo karyokinesis. Okay, karyokinesis. Is there any student who doesn't know karyokinesis in this class? So, tapetal cells will first undergo karyokinesis, but the thing is that karyokinesis is not followed by cytokinesis. Okay, karyokinesis is not followed by cytokinesis. Got it? So because of that, your tapetal cells are binucleate. Very amazing question. Very good question. So that is the reason why your tapetal cells are binucleate. You all know that your tapetum is mainly for nourishment and these cells are binucleate. Got it? Awesome. Karyokinesis is not followed by cytokinesis, or simple word, only karyokinesis happens. Next question. What would be the ploidy, okay, of the cells of a microspore tetrad? All of you tell me in the chat box. See, your MMC, right, your microspore mother cell will undergo division and form your microspore, 4 microspore tetrads. What will be the ploidy of the 4 microspore tetrads? N, N, N and N, right? So, it is going to be haploid or diploid? Haploid or diploid? Your microspore tetrad, that is the ploidy of your cells, will be N. Simple? Yes, awesome. Haploid. Awesome, awesome. Okay, now next question. Why do you think the exine should be hard? What is the function of a germ pore? Okay, that's the question. Why do you think the exine should be hard? The simplest question is, exine is made up of the most hardest thing that is called as sporopollenin. Sporopollenin, and sporopollenin is the most hard material because this has to withstand strong acid, alkali, extreme temperatures. See, because pollen grains can be transferred anywhere, anytime, because the fauna is present everywhere, right? And if the sporopollen is not hard, if the exine is not hard, then pollen will get completely damaged. So, to withstand any kind of condition, be it acid, be it alkali, be it strong temperatures, the sporopollenin has to be hard. Very simple question. Now, what is the function of a germ pore? Since I told you sporopollenin is so hard, if there should be formation, okay, if there should be formation of your pollen tube, right, there should be a space. Because sporopollenin is so hard, the pollen tube formation will not happen. So, there is a small pore, or there is a small pore where the sporopollenin is absent, which is called a germ pore, and in this place the pollen tube will emerge from. Okay, the pollen tube will be able to emerge. Got it, students? Easy? Easy? Yes, easy? Okay, got it. Super, super, super. Fine. Okay, so germ pore is a small pore where sporopollen is absent. In that pore, the pollen tube germination or pollen tube emerging will happen. Next question. See, how long do you think the pollen retains viability? Okay, so again the question, the answer is here only. Viability depends on which type of plant it is. Normally it is 30 minutes, but there are also plants which will go for months and years. Now look here. The period for which the pollen grain remains viable is highly variable and to some extent depends on the prevailing temperatures and humidity. The question can also come, what are the factors that affects the viability of the pollen grain? You have to write temperature and humidity. Okay, so in some cereals such as rice, wheat, pollen grain, they lose their viability within 30 minutes. Okay, within 30 minutes. And then you know your Rosalie or your Rolisa, anything, Roliso, so. Rosole or your, however you want to remember, your Leguminosae, Solanaceae and your Rose family, all of them will have more. See here, look here, Rosaceae, Leguminosae and Solanaceae, they maintain their viability for months. This question was asked. Remember, I solved the most expected question, important questions. This question was asked. Okay, so you can remember Rosole make कौन है, something like that is there, no students? Rasoi, something is there. I remember listening to it, but I cannot remember. Okay, next question. The MMC undergoes meiotic division. Okay, I don't think you can see this question, so let's go here. Okay, see, what is the importance of MMC undergoing meiosis? What will be the ploidy of the cells of the nucellus, MMC, the functional megaspore and female gametophyte? This year CBSE question had a very similar question where they asked about antipodal cells, right? So that similar questions are asked every year. So look here. See, why should the MMC undergo meiosis? All of you tell me in the chat box. Very, very easy. Yes, rise, shine and learn. Awesome. Okay. Why should MMC undergo meiosis? Because if MMC or your megaspore mother cell does not undergo meiosis, then the megaspores formed. Even though 3 of them will not become functional, only 1 of them will be functional. This one's ploidy has to be maintained, right? So, megaspore mother cell has to undergo meiosis to maintain the ploidy. To maintain the ploidy. This is the answer. To maintain the ploidy. Now, what will be the ploidy of the cells of nucellus? Nucellus is a covering, right, around, so it will provide the nourishment. Can all of you tell me what is the ploidy of nucellus? MMC, I'm going to tell, it is diploid. Megaspore, mother cell is diploid. Now, the functional megaspore, that means the female gametophyte, right? The female gamete, which is going to become your embryo sac. That will be N. Female gametophyte, that means your embryo sac. I want the first question only answer. Come on, all of you tell me in the chats. All of you tell me in the chats. This also will be N. So, nucellus, MMC, functional megaspore and female gametophyte, what will be the ploidy? Awesome, Preeti. Awesome, Bhavana. Very good, Krishna. Yes, Subhashni, good job. So, it is 2N, right? So, it is 2N. So, what is the answer? Nucellus, 2N, 2N, N and N. Any doubts in this? This will be the ploidy level. Okay, this will be the ploidy level. Okay, next question. Do you think that cleistogamy is advantageous or disadvantageous to the plant? That means, do they have an advantage or a disadvantage to the plants, and what is the reason? Okay, what is the reason? All of you tell me in the chats. Cleistogamous flower is having advantage or disadvantage of not being open, right? Cleistogamous flower, is it an open flower or a closed flower? They keep themselves closed, right? So, is it good or bad? Is it good or bad? All of you. Is it good or bad? Students, 2N, don't tell 4N please. Yes, good or bad? Advantage. Okay. Advantage, all of you are telling? Quickly, advantage. Okay. Only advantage is there, no disadvantage? Okay, so it is both. It has both advantage and disadvantage. Let me tell you about the advantage. It is that they will have a good set of seed, right? Because they are doing self-pollination, they do not have to worry, right? So, they will have a good set of seed, like assured set of seed. Instead of good, we can use the word assured, more relevant word. Assured set of seed. That is the advantage. Assured set of seed, and they will always do self-pollination, so they do not have to worry about pollinators or condition, temperature, unfavorable situation, nothing they have to bother. Self-pollination is happening. Now, disadvantage is that because they are continuously doing self-pollination, inbreeding depression will happen. No one have revised anything. They are telling only advantages there. Inbreeding depression will happen, and will variation happen? No variation will happen, right? No variation will happen. So, the advantage is that they will have an assured set of seed. Okay, good. They will do self-pollination. Even if the condition is favorable, unfavorable, they are not bothered. Now, disadvantage is that because they are doing continuously self-pollination, inbreeding depression will happen, and no variation because again, continued self-pollination. Got it? So, please do not write only advantage. You will go wrong. Okay, look here. The question is 2 down. So, let's take it here. Now, see. Both wind and water pollinated, who are they talking about? Wind and water pollinated flowers are not very colorful and do not produce nectar. Okay, what would be the reason for this? Very simple reason. Floral rewards, right? Some rewards are given from the plants. Plants give some rewards to the insect because the insect is going to do pollination. All of you got this? To do that, we do not have to give this rewards to water and wind pollinated flowers. We only give it to insects. So, insects are the only one who are going to get floral rewards because they are the only one who come for that, right? In the sense, if the flowers are attractive, insects are going to come. If the flowers are having nectar, insect is going to come. So, this floral rewards are not present in wind and water pollinated flowers. Got it? Got it? So much talking is happening in the chat box. Unnecessary talking in the chat box. You are nearing to Neet, and if now also you are not serious and you are coming to a class and sitting and talking, students, I do not know what you people have to think about yourself. You're so nearing to Neet. Okay, and these are all important questions. Many questions have come directly for Neet. Okay, so those who want to talk, please go to WhatsApp, Telegram, any other place and talk. Got it? Other, there are serious students in the class. Yes. So clear? Insects are given floral rewards because they are the one who comes for that, right? They want the reward. But wind and water pollinated, they do not care that much. So that's the main reason. Now, next question. Endosperm development precedes embryo development. Why? Okay. Endosperm development precedes. Can anyone tell me the answer? endosperm development precedes the embryo development? Because very simple reason, endosperm will have to provide the nutrition or the nourishment. They have to provide the nourishment for the developing or for the growing embryo. So obviously this person has to come before this person. So endosperm has to provide nourishment to the embryo. So endosperm will be preceding the development of embryo. Got it? Awesome. Fine. Next question. Any relationship between number of ovules in an ovary and the number of seeds present in a fruit? Okay, look here. Is there any relationship between the number of... Where is the slide changer? Stole it. Very good. I have to go till there to change it. Okay, students, is there any relationship between the number of ovules in an ovary and the number of seeds present in a fruit? Very simple. Ovules develop into what? Ovules after fertilization become seed and ovary become fruit. All of you know basic things this is. Now, after fertilization, how many ovules are there? That many seeds will be formed, right? That is the connection or that is the relationship. So, after fertilization, how many ovules are there? That many seeds will be developed. Got it? That many seeds will be developed. Okay? Yes, very good. Ovules into seed. Very good. Pooja, Krishna, very good. Okay? Now, how many eggs are present in an embryo? Let's take it from here. How many eggs are present in an embryo sac? How many embryo sacs are present in an ovule? How many ovules are present in an ovary? How many ovaries are present in a typical flower? This is like Betty bought some butter, butter was bitter, so Betty bought some better butter to make the butter bitter better. This is like a tongue twister. Okay, this is like a tongue twister. So, how many eggs are present in an embryo sac, students? One egg is present in an embryo sac. How many embryo sacs are present in an ovule? One embryo sac is present in an ovule. How many ovules are present in an ovary? This is completely variable, right? This is completely variable, right? How many ovules are present? Completely variable. Then, how many ovaries are present in a typical flower? The number of ovaries totally depends on how many carpels are present. They can be multicarpellary, right? So, how many ovaries are present in a typical flower totally depends on the number of carpels which are present. Okay. Now, have you seen this one? One second. This is not. No, I have not seen. So that is not your. Okay. Now, what is fruit production without fertilization called? Okay. What is fruit production without fertilization called? Parthenocarpy. Parthenocarpy. So, a fruit formed without fertilization is called parthenocarpy. Got it? Yes. Fine. Now, take out some seeds. No, I'm not going to do that. Okay. Okay, this is not. No, this is not important. Take out some seeds of orange and squeeze them. Observe how many different. No, this is experimental biology. That's all.
Students, are you ready to do the human reproduction now? Yes. It's nice pen, no?
Okay.
Thank you. Now let's quickly go through over the human reproduction. All of you know this is a very easy chapter and we can do every single question very easily. Starting off with the first question. That's going to be from here. Okay. With the first question, that is, what would be the number of chromosomes in a spermatid? So all of you know spermatid is the result of the meiosis. So it will be haploid in nature. So quickly tell me in the chat, what is the ploidy level is going to be haploid. So the number of chromosomes is going to be 23 pair of 23 chromosomes. Clear? Next question. Does the first polar body born out of the first meiotic division divide further or degenerate? So all of us know the first polar body is the result of your meiosis. When the meiosis 1 is finally completed, which will result in the second polar body as well as second polar body as well as the first polar body as well as your secondary oocyte. Now this secondary oocyte, right, secondary oocyte will undergo first polar body and the primary oocyte, sorry, for the primary oocyte, and this particular secondary first polar body will not go any further. Khel khatam, then it will degenerate completely. Okay. Next question. Can you identify the major differences between your spermatogenesis and oogenesis? Now tell me in the chat, when does the oogenesis start? All of you know the oogenesis starts at the embryonic stage, while the spermatogenesis starts at the puberty. First difference. The second difference is going to be spermatogenesis is a continuous process. Yes. What about oogenesis? Are there any pauses in your oogenesis? The answer is yes. At prophase 1, at diploidine stage, it stops. And after in the meiosis 2 also, does it stop at your metaphase? Yes. So it is a discontinuous process. In the case of your spermatogenesis, how many sperms are produced from your one spermatogonia? 4 sperms. But in the case of your oogenesis, a single secondary oocyte is produced. Yes, the difference done. Next question, students. What is the easy question? How many chromosomes will be there in the zygote? You should tell me in the chat. Remember, students, zygote is diploid in nature. So tell me in the chat, how many chromosomes? Easy question, all of you know. Next question. It's going to be, what are the major features of embryonic development at various months of pregnancy? Students, this particular paragraph right here has so many questions, many questions. Starting with the first month. At the first month, we can see here, month 1, embryo heart is formed. So students, I've told you before, write down here. First month, what happens? Formation of heart, right? The first sign of growing fetus may be noticed by listening to the heart sound carefully through the stethoscope. By the end of the second month of pregnancy, the fetus develops limbs and digits. So second month is going to be what? Limbs and digits. Limbs and digits. At the end of 12 weeks, of first trimester, there is most of the major organ systems are formed. For example, limbs and external genitalia, organs are well developed, need PYQ. The first movement, we say, right, the kick of the baby. Yes, the baby is kicking. That is at the 5th month. The first movement of the fetus and appearance of hair on the head are usually observed during the 5th month. Again, a question, important question. So 5th month, the baby is playing inside already. Next question. By the end of about the 24 weeks, that is end of your second trimester, the body is covered by fine hair and eyelids separate and ashes are formed. And by the end of 9th month, the baby is released outside. I'm saying released outside. Baby is born. Okay. Clear? Students, I'm telling you, please write down this entire, what happens in every week, what happens in every month wise. Question will definitely come from there. Next question, students. Infant is delivered. The placenta is also expelled out of the uterus. What do you think the doctor inject to induce the delivery? So all of you know there is one hormone called as oxytocin is responsible for your delivery. One of the major hormones. If there is not enough of oxytocin, a particular doctor will inject the oxytocin. That is, artificial oxytocin is injected. Clear? Next, we have questions from your principles of inheritance, starting with the first question. That is going to be what he concluded, that is Mendel concluded that genotype of dwarf was having homozygous small t. What do you think he would have got if he had self-pollinated the tall plants? Now imagine, tell me students, if a plant is said to be tall plant, it can be homozygous tall or it can be homozygous tall and it can be heterozygous tall. If we cross homozygous tall into homozygous tall, what do we obtain? We obtain 100% tall plants. Yes. Now what if we cross heterozygous tall and crossing with heterozygous tall, we obtain 75% tall plants. 75% tall plants. Easy question. Next question. Using Punnett square, try to find out the nature of offspring of test cross what ratio did you get? Using the genotype of this cross, can you give the general definition of a test cross? Now, what is a test cross, students? Remember, test cross is a type of your back cross. Yes, test cross is a type of your back cross in which we take a particular phenotypic dominant character and we cross it with your double reciparents, and the genotypic ratio and the phenotypic ratio is going to be 1:1 here. It is going to be 1:1. 1:1. Remember, students, in a test cross, your genotypic ratio is equal to your phenotypic ratio, PR. Genotypic ratio is equal to your phenotypic ratio here. Now, that is the test cross done. Did you understand this? Next question. How many phenotypes are possible? Now, if you have attended my lecture on your class principles of inheritance, the formula to identify the phenotype is going to be, the formula for phenotype is n+1. N is the number of alleles. N is the number of alleles. And remember, in the case of your ABO blood grouping, it is a type of your multiple allelism. Multiple allelism. Now, what is a multiple allelism? In multiple allelism, one gene has many alleles. One gene has many alleles. Now, in the case of your ABO blood grouping, how many alleles do we have? We have 3 alleles. 3 alleles are there. So, if the 3 alleles is there, so N is going to be 3. So, what is the phenotypic ratio? The number of phenotypes which you can obtain is 4. The phenotype which you can obtain is 4, n+1. Now, here we knew it is 3 alleles. So, if in your examination, if they give you hypothetically, if they give you an examination, 10 alleles, what is the phenotype then? Tell me in the chat. Tell me in the chat. In your examination, if they give you 10 alleles, if one gene has 10 alleles, how many phenotype would you obtain? Tell me in the chat. Easy. Next question. How can you tell which of the character in a pair, yellow and green color and round and wrinkled shape, is dominant? The best way to identify this is doing the actual cross by planting. Then you will identify which particular phenotype is obtained in your offspring. Between your yellow and green, yellow is dominant. All of you know. Between round and wrinkle, round is more dominant. Next question. We have given table A, table B. Can you tell which of these combination columns, column A or column B, represents the chromosome and which represents the gene? Students, column A represents your chromosomes and column B represents your gene. First two definitions are almost similar, but if you look at the last one here, independent pair of chromosomes segregate independently of each other. A pair of genes segregate independently of another pair. Law of independent assortment. Are you noticing? That is your answer here. Next question. An X chromosome has become, okay. Do you think the number of chromosomes in the male and female are equal? Context. The context is going to be your insect here. Students, remember, in the case of your insect, we have XX and XO type. Yes, XX and XO type. And here in the case of XO type, will it have same number of chromosomes as the XX? No, because we have XO here, means one chromosome is missing. So male and female will not have the same chromosome here. So male will have one chromosome less. Male will have one chromosome less here. Next question. How is the sex determination mechanism different in the birds? Is the sperm or the egg responsible for the sex of the chicks? Students, remember, in the case of your sex determination, in the case of birth, it's going to be ZW and ZZ. ZZ is going to be your male and ZW is going to be your female, and the sex determination is done with the help of what? The sex determination is done with the help of the egg. Done? Clear? Clear? Done? That is the end of your principles of inheritance. Ma'am will be doing molecular basis now.
Do you have any doubts? Okay, students, let's start by molecular basis of inheritance. First question, compare the structure of purines and pyrimidines. Can you find out why the distance between two polynucleotide chains in a DNA remains constant? Very simple question. Purines are larger, right? They are double ring structure, 2 rings, and pyrimidines are single ring. And between the purines and pyrimidines, there are hydrogen bonds. So these hydrogen bonds allow them to be constant. Very simple question, right? Purines 2 ring, pyrimidines 1 ring. Both of them have hydrogen bond, 2 hydrogen bond and 3 hydrogen bond, and because of that the structure is constant. Okay? Okay? I'm not seeing so much love, okay, when I'm coming back. I'm noticing all that. You see, I'll give you people punishment next class. Okay, so yes, look here, in some virus the flow of information is in reverse direction, that is from RNA to DNA. Can you suggest a simple name of the process? I will not tell the answer, all of you will tell me the answer. Such an easy question. Okay, in some virus the flow of information is reverse, so it is, that means what? From RNA to DNA. Tell me what is that, which is that simple name of the process. Now next question, if the length of E. coli DNA is 1.36 mm, can you calculate the number of base pairs in an E. coli? This I had done in the most repeated question, but let's do it once again. Very good. Come in. Yes, awesome. Okay, students, so look here. Yes, reverse transcription, right? Reverse transcription, the enzyme uses reverse transcriptase. Now look here, all of you, here the question is length of DNA. Okay, length of DNA can be calculated with number of, the question is number of base pairs. Okay, number of base pairs into the distance between two base pairs. Okay, distance between two base pairs. This is how you will calculate it. Now we know length of DNA is 1.36 mm is equal to, let's put it as x into, one second, into 0.34 into 10 to the power minus 9, right? Am I right or wrong? All of you tell me in the chats. Okay, now I have to calculate x. So x will be, shall I write it here? X is equal to, how much is it? Just have to calculate this. Very simple. 0.34 into 10 to the power minus 9 divided by 1.36. If you want mm, you can convert it to nanometer into 10 to the power minus 6, or you can just keep it like this. Answer will be somewhere between, I think, 4.6 into 10 to the power 6 minus 6. Just check this answer. It will be somewhere 4, right? 4. All of you tell me in the chats. Yes? Very good. 4.6 into 10 to the power 6. Awesome. Many of you got the answer. Very good. Very good. Now, look here. How many such beads do you imagine are present in a mammalian cell? What are they talking about? The nucleosomes. How many nucleosomes will be there? That is the question. I hope all of you got the answer. There is no confusion, right? So students, again, I had told you this. One nucleosome, okay, one nucleosome has around 200 base pairs. 200 base pairs. Mammalian cells has how much? 6 into 10 to the power minus 9. Okay, so we have to find this. How will you find it? Simple division. I don't like doing maths, but look what I am supposed to do. Okay, simple. 1 divided by 200 into 6 into 10 to the power minus 9. You can do with unitary method. 3 2s are 6, no? 100. Somewhere you will get into 3 into 10 to the power 7, I believe. Check this also once again. I am giving you maths. Check this. Understood? One nucleosome, 200 base pairs. So 6 into 10 to the power minus 9, our total number of cells. How many nucleosomes will they have? they'll have 1 divided by 200 into 6 into 10 to the power minus 9. With unitary method, if you do, you'll get somewhere in 3 into 10 to the power 7, I believe. Okay, check in, let me know. Yes, 6.6 into 10 to the power minus 9. Right now, I've taken 6 to make it more easier, because there you will not be able to do all of it. But just you keep it 6 also, no problem. The answer should be how much? Yes, awesome, awesome. Very good, very good. 3 into 10 to the power 7. Yes, I'm correct. Wow. My maths teacher is super proud. Okay, can you think of any difference between DNAs and DNA and DNase? Very simple. DNA is the genetic material. DNase is the enzyme that will degrade this genetic material. Okay, and here, you know, difference between DNase and DNA. First thing is genetic material. Second thing is this is an enzyme that will degrade the genetic material. Okay, is there any other difference? Is there any other difference? Think and tell me the difference. Okay. Yes, yes, minus 7 only, fine. Yes. Since I know it is 6.6, keep it 6 and calculate. No, what is the problem? Why is it so difficult to make it easy? Make it easy in life. You keep 6.6 and calculate, no problem. I just taught you the formula. Okay, understood? No, DNA and DNase difference. Next one. Can you recall the two chemical differences between DNA and RNA? Very easy question. DNA and RNA. What is the two chemical differences between DNA and RNA? Main thing is absence and presence of the oxygen group, right? And then you know that DNA is more stable because of the presence of thymine instead of uracil. All this will come under the chemical differences. You can write the differences in purines and pyrimidines. In pyrimidines only, the difference between the presence and absence of oxygen. Got it? Yes, awesome, very good. Yes, DNA is a nucleic acid, DNase is a protein. That also you can write. Now, look here. Can you recall, is it visible the question? I think okay. Can you recall what centrifugal force is and think why a molecule with higher mass or density would sediment faster? I know that centripetal and centrifugal force is there. This is completely physics, but they are moving away from the center. Right, centrifugal force means they move away from the center, and all of you know that centrifugation is done to make sure that there is a gradient difference. Whenever there is a density gradient difference, centrifugation is done. So what happens? The heavier particle will settle in the bottom and the lighter particle will be on the top. The heavier particle will be called the pellet and the lighter particle will be called the supernatant. Got it? Simple. Okay, students, rest is physics. How much physics I know I have told. Okay, rest you please ask Shreyas sir, what is centrifugal force? What do you think about it? Okay, yes. Now, next question. If E. coli was allowed to grow for 80 minutes, okay, 80 minutes, then what would be the proportion of light and hybrid density DNA molecule? See, one generation, okay, one generation will take 20 minutes. Right, one generation will take 20 minutes. So, 80 minutes means we will take 4 generations. 4 generations can be formed in 80 minutes. Okay, can all of you try to find it out? Let's, it will take some time, but let's do it. See, first generation is going to be hybrid, right, because they will be dipping it in heavy and then light. So, say one will be hybrid. Let's draw it like this. First generation hybrids will get. So, I'm writing it here. First gen only hybrids. Okay, 100% hybrids. We have to calculate the percentage. Okay. Second generation, what do you expect? We will get 50% hybrid, 50% light. Agreed? 50% hybrid, 50% light. Third generation, we will get 25% hybrid and 75% light. You can all do this and try. You can all do this and try. Okay. You will be able to get the answer. All of you try this. Try all of you. And then last generation, that the fourth generation, we will get, if you see exactly, you will get 13 hybrid and 86, what is that? Light. But to make the value, it is always 12.5% hybrid, okay, and 85.5% light. Okay, if you want the exact number, it is around 13.3, okay, 13.3 light and 86.6 is, 13.3 is hybrid and 18.6 is light. Students, if you do this way also, you will get the exact number. Okay, any doubts in this? Please let me know right now. Yes, 87, no? Yeah, 87.5. Okay. All of you got it? Yes, awesome, awesome. So that is, see, so first generation 100% hybrid, second generation 50 hybrid, 50 light. Okay, if you take this also separately, you will understand, no, how to do it. Then again, third generation 25 hybrid, 2 hybrid strands, rest all will be light. Here also you will get 2 hybrid strands, rest all will be light. Okay, try and do it. Try it in your book later. If you are having any doubt, please ask me later. Okay, please ask me later. Chalo. Can you now write the sequence of RNA transcribed from the above DNA? This we have already done in our most repeated questions. Okay. Now, do you face any difficulty in predicting the opposite of this one's opposite? If you find any difficulty, the question is here, students. Look here. We have meth, phenyl, and then continuously. Do you face any difficulty in predicting the opposite? Can you now correlate which two properties of genetic code you have learned? Very simple. First is, genetic code is unambiguous. That means what? One code codes for only one amino acid. Second most important characteristic of genetic code is genetic code degenerates. That means some amino acids are coded by more than one codon. Got it? So these two factors are the ones that actually, affect your, the question is what here? Sorry, one second. See, can you now correlate which two properties of genetic code have you learnt? Okay, have you learnt? So that is unambiguous and degenerative. These are the two genetic code characteristics. Is this clear? Yes, AUG, UUU, UUU and UUU. Correct, correct. Okay, methionine, this one has dual, right? AUG, right? This one UUU and it goes on. Got it? Awesome. Now, next question. Now here. Remember, glucose or galactose cannot act as an inducer for the lac operon. Can you think of how long the lac operon would be expressed in the presence of lactose? All of you know, if lactose is present, that means the inducer will go and bind with the repressor and not go and allow the repressor to go bind with the operator, and RNA polymerase will continue translation and transcription. This is basic understanding. The question is, how long will this lac operon run? Lac operon will run until lactose is available. Okay? Until lactose is available for the E. coli, the lac operon concept or the lac operon model will keep continuing. Okay? And RNA polymerase will be happily doing transcription and translation. Simple question. Okay? Now, see here there is a question. Assuming human genome has 3 into 10 to the power 9 base pair, in how many base sequence there would be a difference? All of you know that we all, okay, 99.9% we have same bulk DNA. Okay, only 0.1% is different. So the question is, how many will be different? So if I have to calculate how many will be different, very simple, 0.1% into 10 to the power 9. Okay, this will be my, how to do the calculation. This will be how much, students? 0.001 into 3 into 10 to the power 9. Oh my God, so much maths. That is 1 divided by 1000 to make it convert. Okay, 1 into 1000. Sorry, this into 1000. Oh. So what will be the answer? So if I have to make this 1, what will I do? I will multiply into 1000, right? So I'll get 1. Multiply into 1000. So 1 into 10 to the power 3 into 3 into 10 to the power 9. I think minus sign is missed here, no? No, minus they have not given. So what will be the answer, students? 3 into 10 to the power, supposed to be, this will be minus, so 7. Correct? 3 into 10 to the power 7. Am I right or wrong? All of you tell me. 9 minus 3 is 6. Sorry. 3 into 10 to the power 6. Perfect? Okay, good. So, 0.1% will be different. Okay? Evolution. Okay, very simple question. Do you remember, ready? Evolution. Do you remember how the ages of fossils are calculated? Ages of fossil can be calculated by relative position. That means where this fossil is being there. This I taught you in class. Right? If a fossil is, see, imagine one fossil is here and one fossil is here. All of you know that this B fossil is the oldest fossil because it is more below. Right? B fossil is more below. That means that is more older. Okay, that is one way of understanding the fossil age. Second way is by carbon dating. Second way is by carbon dating, that is taking your C12 isotope or C14, right? Taking any of this and doing carbon dating. Again, I told you one more. Uranium, right? Uranium gets converted to lead. So, if uranium content is more, that means this fossil is little more younger fossil, right? It is not very old, it is very recent fossil. But if it has already become lead, that means it's a more old fossil. All of you remember this? Yes, awesome. Carbon dating and your relative positioning. Okay, do you recollect? I don't recollect. Principle behind it. Okay, so yes, that's all. One question only, evolution. All of you know what is carbon dating? I think all of you know. Now, human health and diseases. First question. Do you know why mother's milk is considered very essential for the newborn baby? That is, your colostrum is very essential for the newborn baby because this is a passive immunity. Passive immunity, right? So, this passive immunity provides what for the child? So easy. It provides IgA. To provide this IgA, colostrum is very necessary. That is why the first milk of the mother is very important for the baby. Awesome. IgA. Next question. Here, let's look here. When do people need repeated blood transfusion? Find out and make a list of these conditions. Okay. When does the person need continuously blood transfusion and what is the need for it? Yes, very good. Natural passive immunity. Now, when do people need continuous blood transfusion? Especially when there is a very severe injury, right? When there is a severe injury or there is a very severe surgery happening, this person will need excessive blood. Or if the person have like conditions like thalassemia, what did I say? Okay, if the person have sickle cell anemia, or if the person has any anemic condition, like if there is a kidney failure or if there is any problem with the kidney, lungs, any organs and that is anemic, that means loss of blood is happening, the person will need continuous blood transfusion. Is this clear? Yes, hemophilia, very good. Hemophilia is there, thalassemia is there, leukemia is there. Leukemia we'll write. Leukemia. Okay, during all these conditions, a person will have to be continuously given blood transfusion. Got it? Yes, very good. Now, microbes and human welfare. First question. Do you remember which kingdoms among the living organisms contain microorganisms? Such a baby question, students. Come on, all of you can answer. Obviously, it is Monera, Protista. Okay, in this Protista, you have amoeba, example. And you also have fungi. Fungi also only example is yeast. These are all microscopic in nature and these are the kingdoms that fall into it, right? Monera, Protista, fungi. Fungi may yeast. Don't forget. Okay. Now, next question. Can you tell which metabolic pathway is taking place resulting in the formation of carbon dioxide? Obviously, the metabolic pathway has to be anaerobic respiration, right? Anaerobic, that is anaerobic fermentation. This anaerobic fermentation can lead to two types. One is alcoholic and one is lactic acid, right? One is alcoholic, one is lactic acid fermentation. So, this will lead to the formation of carbon dioxide. So, if you ferment something, release of carbon dioxide will happen, okay? Yes, Monera Protista. Yes, fermentation. Sorry, I was listening to Sir's respiration, so it became respiration by mistake. Okay, so
This is the impact.
Nothing like that, students. Anaerobic fermentation. Okay. Next question. Do you recollect the metabolic reaction which results in the production of ethanol? Again, same question. Fermentation you write and you write specifically alcoholic fermentation. That's all. Specifically alcoholic fermentation. Lactic acid also happens in our body. Okay. I forgot fermentation spelling suddenly. Yes, alcoholic fermentation. Okay. Done. Done. Yes, done. Okay. Next question, something I have asked. Do you think we human beings are able to digest the cellulose present in our food? Of course not. No. Otherwise, we would be eating grass like Sir told. Plants are very intelligent. From tomorrow, Sir is going to do photosynthesis and now he is eating biscuit. Go stand in sun. You'll become more intelligent. Go do photosynthesis and come. Okay, students, do you think we humans can digest? From tomorrow we will get to know.
I know even you want biscuit, that's why, right?
That's not, I'm anyway, I'm ready to be an animal with little brain cells damaged also, but you want to do plants, okay? So human beings are able? No. No is the answer because we do not have cellulase to digest the cellulose. It is only present in cows and cattle, right? It is present in cattle. So they have cellulase which will break down the cellulose, and that's the reason why they consume plants and they can break it down, okay? Yes, we do not have, okay? But Baswa sir has. Baswa-sauruses, as the name suggests.
It's an animal,
but he's now converting into plant, students. Okay, from today onwards, we'll call him plant. Okay, can you tell what advantage the fungus derives from this association? Which association? Association. Association, students. Okay, they are talking about symbiotic relationship. That means your mycorrhiza. Okay, so what is that? That means both of them are benefited. Who are both of them? This fungi will be present on the root nodules of the plants, and what is going to happen? They are going to get continuous intake of carbohydrate. That means food. Okay, so only thing the fungus has to do is convert this nitrogenous atmospheric nitrogen into a nitrogen and increase the fertility of the soil, right? But then these people are continuously getting carbohydrate and energy from the plant. Okay, clear? Yes. Okay, very good. Okay, symbiotic relationship. Here they are talking about mycorrhiza. Okay, not the lichen, not the algae fungi. Here the plant and fungi relation. Okay, plant and fungi relation. Here you can see many members of glomus. See, what are the benefits? Such as the resistant to root-borne pathogens. This is the benefit that fungus is giving. So, yeah, exactly. The most important thing is they will be provided with energy, right? Carbohydrate will be provided for them. Now, biotechnology, sir. Principles. I'll only do students, both the biotechnology. No, I'll do both. Okay, let's do biotechnology, principles and processes. Okay, first question here. Since DNA is hydrophilic molecule, it cannot pass through cell membrane. Why? Because the cell membrane or the plasma membrane is hydrophobic in nature, and this DNA is hydrophilic. So, hydrophilic, that is your water loving people cannot just directly enter. They will need some people's help to enter. Okay. So, that is the reason why this DNA will not be allowed inside, mainly because the plasma membrane is hydrophobic in nature. Got it? They fear water. Okay, they are hydrophobic in nature, and directly hydrophilic substances cannot enter inside. Yes or no? Yes or no? Biotechnology. All are crying. So happy they are seeing biotechnology. Next question. Look here. After having cloned the gene. Okay, let's look here. Okay, look here. After having cloned the gene of interest and having optimized the condition to reduce the expression of the target protein, one has to consider producing it on large scale. Can you think of any reason why there is a need for large scale production? If any protein encoding gene is expressed in a heterogeneous, all that is not needed. Students, basically, if you are doing something, right? See, here the answer is actually there. See, I don't know if it will be visible. Wait, let me try to zoom and show students. Here, it's too big, no? Yeah, look here. The question is, the answer is given here actually. See, if there is, the cells harboring cloned genes of interest may be grown on a small scale in the laboratory. The culture may be used for extracting the desired protein and then purifying it by using different separation technique. Students, when you are doing something of a desired quality and you want to make it into large scale, it is always better that you start with a small scale. But if you need something for commercialization, that means you want to export it, you want to sell it, then you have to do it large scale, and there you can use your bioreactors and you can use big technologies to make it large scale. This is actually not a question as such, but this is just like, can we do a large scale? Yes, you can do a large scale. Okay. Chalo. What would a diabetic patient do if enough human insulin was not available? What would a person do if there is no enough insulin in their body? Basically, they will take injections, right? We will start taking it from outside. 1 second. What is happening? What is happening? It's not working, yeah? Yes. Basically, that person has to take injection. The person has to take injection from outside. Am I too fast? Is it fine? I think it's fine, right? Okay, so would the insulin isolated from other animals be just as effective as that secreted by the human body itself? And would it not elicit the immune response of a human? So what happens? Our body is not making enough insulin, and we have also studied this in your chemical coordination also, right? So what is going to happen? Imagine I take insulin from a pig or any other animal example, or a cattle. Now that can cause allergy in my body, right? An allergic response could be caused. It is not necessary, but there are chances that an allergic response can be caused because I'm taking it from another animal. Agreed, all of you? All of you tell me in the chats yes or no. 2 minutes, I will wait. Yes or no? If I am taking an insulin extracted from any other animal. Yeah, that's what I told. Pig and cattle, imagine. Then, is there a chance for me to have an allergy? Yes or no? Yes, allergy can happen, right? Allergy can be happening. That is, your IgE will be released. Again, that is to do with your immune response. But understand here, allergy can be released. So, this is one impact of that. Okay, next. Think about whether insulin can be orally administered to a diabetic people or not. Now, again in chemical coordination, we studied that insulin is a peptide hormone. Insulin is a peptide hormone. And we all know in our body there is a person sitting to degrade the protein. You can tell it as proteinaceous hormone or you tell peptide hormone, easy way. There is a person in our body to degrade protein. Who is that person? Who is that person? All of you in the chats. There's a person sitting to degrade the protein. So, if I orally take insulin, then in the stomach, this is going to get degraded and the effect is not going to be used, right? This is going to get degraded in my stomach because there is protease which will degrade this peptide hormone and insulin, okay, peptide hormone or a peptide proteinaceous hormone which will get degraded and then the effect is useless, right? So, you should not orally take, yes, very good, protease. You should not orally take insulin. Insulin should always be injected. Easy question, that's all. Organism and population. Yes, this also I will be doing, students. Okay, let's start.
So, with that being said, Gopika ma'am, once
part is over in this beautiful journey of life.
Blah, 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 blah. You, as if you're telling you're going somewhere else. So, students, can we start with organism and population right now? Let's have the first question, ma'am. Pen.
Take.
Nothing, you take it, everything and go. Huh? Yes. Let's have the first question from your organism and population. That is, why are night blooming flowers? Why are the night blooming flowers, what is the red color? Night blooming flowers generally white in color. They're white in color because they can, your honeybees and as well as other insects can easily identify them. Obviously, white will be more visible in the darkness. Logic, plants are using logic here. How does the bee know which flower it has nectar? There are different electrical signals. With the help of electrical signals, a particular insect is able to sense this particular flower has nectar in it, hidden nectar. Next question. Why does the cactus have so many thorns? Tell me in the chat, why does the cactus have so many thorns? Why is it requirement? Students, the main reason the cactus has so many thorns is because protection. They're intelligent. Yes, they're intelligent. That's why. Thorns are very intelligent. Unlike your stupid animals, they did not develop anything. Plants, cactus is so intelligent. It is reducing its leaf. There are two reasons. First one is going to be protection. The other reason is reducing the transpiration loss. The plant is so intelligent that a particular environment is so hot, it knows that if it has a broad leaf, all the water will evaporate through the stomata. So what does it have? It has shrunken stomata and reduced to spines. Smart plants, bee-like plants. How does the chick Spurs recognizes her own mother? That is called as imprinting. You might have seen in Tom and Jerry, no? The duck as it saw the Tom, it thought the Tom or Jerry. The Jerry is the mother. That is called as imprinting. In Tom?
It's still not become a plant.
See, plants are smart. I didn't talk about memory there. Next question, students. If you have a dense laboratory culture of bacteria, Imagine all of your scientists now, you have dense very much of bacteria given to you. Okay, in a petri dish, this is very important. If this was a broth culture, then you could have done a method called as turbidity test. There are two different things. Broth is liquid. Petri dish, what we make on petri dish is going to be made with the help of agar. So this is going to be solid in nature. So we have a petri dish here. Nice circle, no? We have a petri dish here. Now on this petri dish, imagine there is a bacteria growing everywhere. Now how do you count the number of bacteria? Very simple. When we were asked this same question in MSc, we were shocked. Like, how do you count? Do you sit and count the entire thing? Then our teacher told us there's a method called as quadrant method. So what do is you divide the entire petri dish into 4 quarters and you count the number of bacteria in one particular quadrant. You count the bacteria here. How much bacteria is there? Then you do multiply it into 4 times and you get the entire bacterial growth colony. The other methods are going to be turbidity test, and there is other method for your you take a sample there and check. So there are multiple methods. This is the most efficient method. Next question. What would happen if all the energy fixed by the autotrophic organisms if the community has no animals to eat? So imagine if this is a food chain, we have plants, grasshopper, frog. Yes, this is a simple food chain. Now in this food chain, what if there is no grasshopper? What if there is no cow? What if there is no goat? What if there is no humans? There is no particular herbivores. What will happen to the plants? Plants are very smart. They will continuously grow now. So plants population will skyrocket. Yes, the plants population will skyrocket. If the plants population is skyrocketing, will there be any conflict now? Imagine there are 2 plants that are growing simultaneously. Will the 2 plants be able to survive together using the same resource for a very long time? No. So if there is a population increase, can I tell competition is also increasing? Just like a NEET examination, population is increasing, competition is also increasing. That is the answer. Next question, students. Do you believe that an ideal parasite should be able to thrive within the host without harming it? No. A parasite by definition itself is harming. If it is not harming, it will become commensalism, right? Then how is a parasite? Well, how will it be a parasite then? Should have some identity, no? So that identity here is it is be harming. Next. There's no question here only. The female mosquito is not considered a parasite, although it needs our blood for reproduction.
Not considered a parasite.
Parasite. That is, it is only entering the bloodstream for reproduction purposes, but for completing the life cycle. That's it. Rest you need to know the entire life cycle of your malaria.
Taking a blood meal.
Blood meal. That is female plus mosquito is coming and sucking the blood. The male mosquito doesn't bite there. That I remember. Useless it is. Why will it even bite? Doesn't even carry eggs. Next question. Measurement of biomass in terms of dry weight is more accurate. Why? Do tell me in the chat. Why is dry weight more important than wet weight? Students, remember the wet weight can be influenced by physiological and environmental factors. But in the case of dry weight, no, there is no factors involved, affecting it. Water can evaporate and wet biomass can change, but dry biomass will not change, and hence we take the dry biomass everywhere. Okay. Now, next question is here. Carnivores, secondary carnivores in the grazing food chain. Do you think there is such limitations in detritus food chain? The answer is yes, there are limitations, because students, in your detritus food chain, is there any source of sunlight there? No. There is no particular primary producer there. And in fact, in your detritus food chain, a particular organism can enter in many different trophic levels. So there are many limitations when it comes to your detritus food chain. Next question, students. Secondary consumers, when it eats insects and worms, can you work out how many trophic levels human beings function at a food chain? Answer is many. If you draw any type of food chain, humans will be there, like in the top consumers. Next question. These 2 questions are very important. Both of them are NEET PYQ. What is the question here? There are exceptions to this generalization. That is, pyramid of number is upright. but the pyramid of number can also be inverted. Example is here. If you were to count, see pyramid of number, count the number of insects feeding on a big tree. So imagine we have one big tree here. On this one big tree, there are many insects feeding on. One big tree, many insects are feeding on that one big tree. What kind of pyramid would you get? Now add an estimate of number of small birds depending upon the insects. So on this large group of insects, there are some birds also there which are feeding on it. So when it comes to pyramid of number, listen to me very carefully, all of you. When it comes to pyramid of number, in your normal ecosystem, grassland, it is upright. But pyramid of number can also be inverted in the case of a tree example. Questions will come from here. Similarly, the pyramid of biomass is generally upright in your terrestrial ecosystem. The same pyramid of biomass is inverted when it comes to the aquatic ecosystem, that is ocean example. See here, the pyramid of biomass in sea is generally inverted. Neet PYQ, I don't know how many times this question has come, because the biomass of fishes far exceeds the phytoplankton. Isn't that a paradox? How would you explain this? Very simple explanation. Listen to me very carefully. The pyramid of biomass is inverted because the biomass of phytoplanktons is actually comparatively less. Why? There are two main reasons here. The biomass of phytoplanktons is less. First one is because the phytoplanktons are very tiny compared to fishes. They're microscopic in nature. So biomass will be definitely less. The second main reason is that the life cycle of phytoplanktons are very short duration. And by the time they finish their life cycle, it takes very less time. So biomass will also be less. So students, in the case of your ocean ecosystem, the biomass of your phytoplanktons is less compared to your fishes, and that is why pyramid of biomass is inverted. Clear? Pyramid of biomass is inverted. Now, next question, students. Last chapter here. What is so special about tropics that might account for their great biological diversity? Remember, students, in the case of your tropics, there is equator. In the equator, the tropics region, is there a high sunlight region? If there is high sunlight, tell me, if there is sunlight is very high, will the plants be able to do more photosynthesis? If there is more photosynthesis, there are more number of food chains. Yes, if there are more number of food chains, it means there are technically higher biodiversity there. That is the answer. High biodiversity. Next question, students. I believe this is the last question here. What do a stream, this is your species area relationship. Species area relationship tells you that the more you search, the more species you will find, but only up to a certain limit. We have an equation also. What is the equation? Log S is equal to log C plus Z log A. Log S is equal to be species richness. Log C is going to be your y-intercept. Z is going to be slope. And A is going to be area. Now students, this Z is called as your slope. Now this slope can become much more steeper in nature. That is, what do a steeper slope means in this context? The meaning is very simple. It means the Z value is very high. The meaning is that species richness is also very high. Simple. That is, if the Z value is high, species richness will also be high. And when do you notice this? Steeper slopes. There it is. The entire chapter is done. Entire chapter is done. Done. Entire, every single chapter of your class 11th and 12th, every single chapter of your class 11th and 12th in text questions are done. In text questions are done now, students. Now what you need to do, students? There are 2 or 3 things which you need to do. First thing is going to be, ma'am, first thing is going to be like the video more.
Yes, like the video and share the video with your friends because in-text questions are mostly ignored by most of the students.
And you have not ignored it now. You have not ignored it. Now you know the importance of in-text questions and how many PYQs have actually come from your in-text questions. Yes. And we'll be doing a similar videos for your bold letters. We'll show you where the questions have come, bracket, disorders, scientist name. So we need to make our students completely perfect in your NCERT, right? And
yes.
Other way to perfect is going to be the crash course, ma'am.
Yes. So students, click on this link. You will be opening into the crash course. That is, look here, view course plan. So what are the availabilities of this beautiful
students?
crash course?
Trust me, if you have not bought it right now, this is the last chance because I think I'll be taking class next week there on the wave. I'll be taking class on this crash course and I'll be doing a...
I'll be taking tomorrow.
Oh, man, I'll be taking tomorrow. See, we'll be taking a course on the crash course and students, trust me, I'll be teaching morphoanatomy in just 1.5 hours. Entire morphology anatomy in 1.5 hours, and you'll know what exactly differences are. Apart from that, you need to know here is going to be your live class by your master teachers. As we are going to be there in live doubt solving. We have dedicated doubt solving counter there. Then we have what? Assignments after class. Then we have smart material, that is Tattva books.
Tattva books.
Right? We'll be giving beautiful books. Beautiful books. Last moment revision. Perfect test for last
We
month.
have used it while our preparation was
Exactly. All India test series, post-class doubt solving, discussion of previous year questions, in-class quizzes and leaderboard. Do you see? For such affordable prices, that is economical, excellence, end game. This is where you go. This is where you go.
1,000 rupees.
No, 999.
Yeah, 1 rupee.
What?
999 students. I always say whenever it's 999, I always say 1,000 or whatever. So here, look here. So this course will be there till the end of your NEET preparation, and during even the paper discussion also, this course will help you because you can download everything. Still, you will have access to everything. Okay. So quickly go join. Tomorrow will be my first class at 10 a.m. Okay, I will be doing human physiology, some concepts, and I will also be doing some problems, PYQs also. So do see, I expect all of you to join because this is the end game. And if you're a student who's not doing proper revision, who is just, attending some lives and doing something, this is your ultimate plan because again, I always say, in the last 30 days, there are a lot of students who changed the entire preparation game by doing proper revision. Okay. And I don't think this is a lot of money. This can be afforded by all the parents and the students who are preparing for NEET. So thank you so much for joining this session. And thanks a lot.
We enjoyed it and hopefully you enjoy it, and let us know in the comment section, students, because we read the comments all the time and it makes us so happy when we read good comments. So let us know in the comment section how was today's class and if it helped you in any way. Right, ma'am?
Yes, and 1 like always comes from us.
Yeah.
Okay.
If you see a like on your comment, right, or 2 likes on your comment, it's going to be us only.
Exactly.
We only like all your comments.
Okay, students. Bye-bye. See you. Happy Sunday. Have fun and quickly join the crash course. Okay, because I will have my class tomorrow and I would love to see all my students there. Ta-ta.
Yes. Bye-bye.